My name is Bob Merison-Hart. I'm from Plymouth University, and my project is about Parkinson's disease and tadpoles. Well, when I began the project, I wanted to use my computational background to try to understand Parkinson's disease. And in order to do that, I started building computational and mathematical models of the parts of the brain affected by Parkinson's disease. The difficulty with that, of course, is that there are billions of neurons in our brain and an astronomical number of connections between them. And so, as kind of a starting point, we thought we need to understand something that is more basic than the human brain. So we studied the spinal cord of the tadpole. And the reason we study that is because it only has about 2,000 neurons in it and 150,000-ish connections between them. And from previous work, we have a fairly good idea of how the neurons are connected. So my job on the project was to develop software that can simulate the electrical and the chemical activity in all of these neurons. And when we do that, we find that the activity patterns that develop when we, say, touch the virtual tadpole is the same as the activity that is seen in experiments with real tadpoles, in that it begins to swim away and generate this swimming pattern of motor activity, which is what you can see visualized on this screen down here. So this is a representation, a 3D representation of the tadpole's spinal cord. Each of these lines is an axon, which is how the neurons are wired to each other. And you'll see that every now and again they're lighting up, and that represents generating a pulse of electrical activity. And what the simulations showed here is that this pattern of swimming activity emerges in response to touch, and it looks very similar to how it looks in real animals. Now I've taken this same software and I've applied it to parts of the rodent brain, actually, which are similar to parts of the human brain affected by Parkinson's disease. So we built what we thought was a good model of a nervous system that behaves like the real animal. And then the question you're probably wondering is, why is this relevant to Parkinson's disease? And the answer to that really is that the neurons in a tadpole are not hugely dissimilar from the neurons in our own brains, in that they're made of the same basic building blocks. They communicate with each other in a similar way. And we believe that the simple circuits that they form in animals like the tadpole, probably those circuits are also present in the human brain. So if we can understand a simple animal, hopefully we can understand something more complicated. So what I did was I took this software that I developed for the tadpole and I implemented a model of the rodent brain and a part of the rodent brain that is affected in rodent models of Parkinson's disease. And I looked at what happens in this very realistic, detailed model of the rodent brain when you go from the healthy case to the Parkinsonian case. And we found, again, in that situation, that the activity that arises in the model is in some way similar to the activity that arises in a real animal in the lab. But the beauty of a computational model is it allows us to look in detail at the mechanisms of how that occurs in ways that you couldn't do in a real animal. We're hoping to take this forward and join it up to a model of a particular treatment for Parkinson's disease called deep brain stimulation, so we can use our computational model to try to unpick why this treatment works and hopefully improve it.